Hello, everybody who is joining us today. I would like to welcome you to this symposium of transposable elements in the era of biodiversity genomics. As you might already know, transposable elements are mobile genomic elements, which are widespread throughout the genome and have been linked to many different evolutionary processes. In the past, studying transposable elements had been quite a challenge, especially due to lack of sufficient quality data because of difficulties in assembling and annotation of transposons. With new long-read sequencing technologies, it has become a lot easier to properly assemble and annotate transposons, and also with large consortiums such as the Tree of Life and other big projects who are generating high-quality genomic resources. It's been possible to revolutionize this exciting field of research on transposable elements. So today, within this symposium, we're going to hear about the work of four early career researchers focusing on analysis of transposable elements in evolution. And I'm going to talk a little bit, introduce each of our speakers, and then we are going to go into going through all of the talks one by one. So our first speaker for today is Marta Coronado Zamora, who is a postdoctoral researcher at Barcelona, working with Josefa Gonzalez group, and is going to talk to us about adaptation in Anopheles mosquito genomes in relation to transposable elements. Our second speaker of the day is Jakob Martellosi, who is currently a postdoc researcher at Senckenberg in Frankfurt, recently joined my research group, but is actually going to talk to us about his previous work in Bologna, from University of Bologna, talking about retrotransposon expansion in truffle genomes using new genomic data. Our third speaker of the day will be Pio Sierra. Pio is a computational genomicist working at the University of Cambridge. He's currently doing his PhD with Richard Durbin's team, and will talk to us about his new tool for annotating transposons in pangenomes and additional analysis on cichlids and Lepidoptera genomes. And then our fourth speaker of the day is going to be Jule Drehalovsky, who is a PhD student at University of Copenhagen, working with Josephine Stiller's team, and is going to show us some new analysis on significant genomes and subsequent genomic expansion due to transposable elements. While the presentations are happening, please feel free to add your questions in the chat, and we are going to have a Q&A session at the end for all the talks together. So maybe we can get started with the talks now.
Good morning, everyone. I am Marta Coronado, and I am a postdoctoral researcher in the Evolutionary and Functional Genomics Lab at the Botanical Institute of Barcelona. First of all, I would like to thank the organizers for giving me the opportunity to present my work at the Transposon Evolution session. Today, I'm going to talk about how transposable elements contribute to Anopheles mosquitoes adaptation in urban environments. Before starting, I would like to mention that this work is in collaboration with Diego Ayala at the ERD. So let's start introducing you to the topic. Anopheles colucci and Anopheles gummy mosquitoes are the most important malaria vectors in sub-Saharan Africa. Indeed, they account for most of the human malaria transmission in this continent. Until recently, urban environments were considered to be unfit for Anopheles larvae development, and it was thought that they will only proliferate in rural areas. However, these mosquito species have rapidly adapted to polluted habitats in the last years, representing a challenge for malaria control. So understanding the genetic factors driving this adaptability of these mosquitoes is very important for a better implementation of vector control strategies and the ultimate goal of eradicating malaria. To be able to understand and to study the adaptation of these two species in urban areas, we have performed a big effort to sample and sequence whole genomes of mosquitoes across several cities from Central Africa, as you can see in the map. Some of the sampling was complemented with public data from the Anopheles gambiae 1000 Genomes Project, in particular to obtain samples from rural areas, because in this work we've mainly provided samples from urban settings, which are in fact more difficult to obtain. So in total, we have analyzed 375 samples, for which more than 70% are new from this project, sequenced for the first time. So by taking advantage also of recent high-quality long-read genome assemblies for both species, in this project we focused on identifying genetic variants from SNPs to transposable elements that could be associated with urban adaptation. And while for the detection of SNPs, there exist gold standard tools and clear protocols, the detection and study of transposable elements is a more challenging task. And probably you already know that if you are interested in working with these. So usually the first task is to have a high quality TE library with all consensus of your species of interest. In the case of Anopheles colucci, in our lab, we already produced a manually curated TE library consisting on 503 TE families. But for Anopheles cambia, we needed to create a TE library as none was available. So for that, taking advantage of this recent long-read genome assembly that I mentioned before, we first used repeat modeler 2 to obtain all possible TE consensus from that genome. But then this raw TE library needs to be manually curated, and this step is very important. So for that, we use a tool that was developed in our lab that is called MC Helper, and here you have the reference to this tool. MC Helper reduced the redundancy of these raw TE libraries and also improves the fragmentations and also the classification of unknown TE sequences and the extension of these consensus. So it reduces the task of manually curate a library for days to just some hours, as it took in my case. So in the end, this Anopheles cambia library consisted on 295 TE families with 53 new ones that were mainly from the mite family. So once we had the library, we can then use tools to detect de novo insertions in our samples. And because these tools can give very different results, we use a combination of 3 different tools. We use Teflon, Population T2, and TEM2, and we only trust those insertions that were detected by the TE programs. So in this figure, you can see the site frequency spectrum of these insertions in Anopheles colucci and Anopheles cambia samples. And although most of the insertions are unique, as we were expecting them to be unique, we highlight here more than 4,700 in Anopheles colucci and 5,400 in Gambia that are present in more than 10% of the samples. They are in high frequency. And indeed, you can see here some of the insertions that they are close to fixation. So in general, we are very interested in these TE insertions because they could be associated with adaptation. And that is why we try to detect the signals of selection, not only in SNPs, but also in transposable elements. And this is what I'm going to show you in this slide. So in this Manhattan plot, I'm showing you the result of running a genome-wide scan of selection using the C2 statistic, the contrast to statistic from the bypass software, comparing urban versus rural samples from Anopheles colucci. To be able to detect with more power regions under selection and to reduce the noise of these scans, I applied the local score approach that here you have the reference. So here in the plot, I highlighted in pink those windows that contain genes that were also detected as outliers when using classic tests for selection, such as the XP-AHH and XP-NSL from the CellScan software. So we are able to detect known candidates, such as the RDL gene, which is associated with resistance to DDT insecticides, and also this cluster of glutathione transferrin genes, which are also related with the metabolism of insecticides. Interestingly, here I highlight also some genes in orange that they are placed nearby a TE with a high frequency. I performed the same analysis, but in this case in Anopheles gambia, and also you can see that we detected some outlier genes that could be associated with the adaptation. But for a better comparison, let's put together the two figures. So now we clearly see that there are some sweeps that are species specific that we only are able to detect in one of the two genomes, but there are other sweeps that they are shared. Like for example, here in chromosome X, that we detected the gene RDG8. And here, as I mentioned before, in both species, we are able to detect some genes that are nearby these with high frequency. So we wanted to investigate a bit more this sequence, and we found that some of these Ds were enriched for insecticide resistance and immunity transcription factor binding site motifs. Meaning that these Ds could be regulating the expression of these genes. So apart from performing classic genome-wide scans for selection, thanks to this sampling performed across different urban and rural areas, we can also run a genotype environment association analysis, or GEM, using as an environmental variable the level of urbanization. And to compute this level of urbanization or urbanization score, we use a tool that allows you to take satellite pictures of a sampling location, and they compute the percentage of buildings, trees or vegetation and roads, and in the end gives you a score of the level of urbanization. So as you can see in this map, we have samples with no urbanization component, like these ones here, that they are taken directly in a forest, and others with a high level of urbanization, as the one that I'm highlighting in the example that was taken from the Douala, from a city. So the idea of this test is to identify genomic regions with allele frequency shifts associated with this level of urbanization. So by performing this gene analysis and keeping only those genes that also exhibit high levels of population differentiation as measured with the XDX statistic, which is very similar to an FST, we identified 203 genes in Anopheles gambia and 83 genes in Anopheles colucci that were associated with this level of urbanization. A few of them, as you can see here, were found nearby these in high frequency, but I'm going to highlight these 13 that they are found in common in both species, suggesting parallel adaptation. And by performing a gene ontology enrichment analysis, we found that these genes were enriched for molecular functions associated with channel activity and transmission and also signaling. So taking together everything, both the contrast analysis and the gene environment association analysis, we are able to identify strong candidate genes involved in this urbanization response. Many of these genes, as you can see here, are highlighted in bold. They are nearby. Is present in high frequency, that I will go in detail into them in a moment. And we also found genes in common in both species. Again, the RDL candidate associated with the DDT resistance, and also the CALX gene. So coming back to these genes that are associated with these present at high frequency, I also perform a gene ontology enrichment analysis, and I found that in the case of Anopheles colucci, these genes are associated with stabilization of membrane potential, and in the case of gambia, on signaling and cell communication processes. So to finish, here you have the take-home messages of my talk. So by combining selective swift scans and gene environment association analysis, we have found known candidates and also new ones associated with the urbanization response, that in the case of Anopheles colucci, they are related to channel transporter activity, and in the case of gambia, with signaling and response to stimulus. That we have generated the first manually curated T library for Anopheles gambia that includes 53 new families, suggesting that more T diversity remains to be discovered when we have new genomes. and finding that these are associated with more than 50% of strong candidate genes related to the urbanization response, suggesting that these play a significant role in this process. So to finish, I would like to thank my group, especially my supervisor, collaborators, and all the people that contribute to this work. And also thank you for your attention, and I am happy to take questions.
Hi, everybody. I'm Jacopo Martellossi. Currently, I'm a postdoc at the Senckenberg Museum in Frankfurt. But today, I will talk about a project that I started during the last year of my PhD at the University of Bologna, about evolution of LTR retrotransposons in true truffles, so in fungi. Nowadays, we all know the pivotal roles that fungi play in the functioning of our planet ecosystem. Indeed, they are core components of nutrients, carbon cycling, as well as carbon sequestration. Moreover, nearly 90% of land plants are found to be in association with one or more fungal partners. Among the most famous association between fungi and plants, we have ectomycorrhizae. So ectomycorrhizal fungi can establish symbiotic relationships with plant roots, where their hyphae colonize the intercellular spaces of the roots, forming what are called hearting nests, that basically are surfaces on which we have exchange of nutrients between the fungi and the plant. Truffles are ectomycorrhizal fungi, but they also have the particular features of forming hypogeous fruiting body. So they rely on mycophagy for spore dispersal. True truffles are the truffles that we usually eat. So they are very famous for their particular fragrance. They are gourmet edible, and they belong to Ascomycota of the tuber genus, so family Tuberaceae. Angiosperms are the main host plants of true truffles, but we can also see some association with Pinaceae. But this is considered to be the derived state. So very likely ancestral truffle species were already colonizing angiosperms roots. Obviously, the particular ecology of truffles leaves some clear genomic signatures. So firstly, we have a restricted set of plant cell wall degrading enzymes. We know that lineage-specific secreted proteins play a pivotal roles in the establishment of ectomycorrhizae, facilitating the communication between the plant and the fungi. And finally, all Tuberaceae species sequenced to date are characterized by very high T content, particularly driven by gypsy elements. But these gypsy expansions are not lineage-specific features of truffles, but is instead, they seems to be like convergent evolutionary features, genomic features of ectomycorrhizal fungi. Here we can see in red all ectomycorrhizal fungi, and we can see that all of them have very high T content and specifically very high gypsy content. However, little is known about the evolution of these gypsy elements as well as their genomic consequences, mainly because of the lack of high-quality genomes. For example, in Tuberaceae, we only have short race-based genome assemblies. We therefore solved this gap, producing a high-quality genome for the Chinese white truffle Tuber pansiwanensis, where we sequence the haploid fruiting body using hi-fi sequencing technology. We managed to get a good representation of its genome with an assembly size of 110 megabases and a contigent 50 of 8 megabases, which is much higher than available publicly available tuber genomes to date. The nuclear genome is included in 28 condigs. of which 11 are composed only by rDNA arrays. And similarly to other ectomycorrhizal fungi, where there is very high T content, around the 67%, where gypsy elements cover the 30% of the genome, so about half of the total T content. When we look at the distribution of genes and transposable elements in the assembly, we found a very strong compartmentalization of the genome, where genomic regions rich in transposable elements are very poor in genes. So genes basically tend to avoid T-rich genomic regions. Indeed, there is a very high negative correlation between gene density and transposon density, and most of the genome is included in T hotspots or T cold spots, where T hotspots are genomic regions significantly enriched in transposons with a mean T content of 91% and very few number of protein coding genes. On the other end, T cold spots cover the 33% of the genome, and here we have very low T content, but very high number of host protein coding genes. So these genomic regions are very dense in genes. We look a little bit deeper at which gypsy clades are hosted in the Chinese white truffle genome, and we found that the three most abundant clades, Prometheus, TMT1, and TCN-like, are all chromovirus-related gypsy elements. So basically, they have a chromodomain in their ORFs. We therefore decided to study a little bit at a more broad taxonomic scale the distribution of these clades along Tuberaceae in particular. And firstly, we performed a novel divergence time for Ascomycota, including Tuberaceae. And we found that this clade originate during the late Cretaceous with the following Paleogene diversification. During this diversification, different gypsy clades expand independently along different truffle lineages. So basically, we have different contribution of different clades to the overall transposons content of Tuberaceae. But the phenotypic endpoint was always the same, so high genome sites. Indeed, we found a very strong positive correlation between assembly sites as a proxy of genome sites and gypsy content, whereas we didn't identify this positive correlation for other transposons groups. The lineage-specific activity of gypsy element was confirmed by phylogenetic analysis. Here, each branch is a gypsy element. Basically, different colors represent different species. We can see that we have species-specific clades, highly dense, and well separated by other clades. We therefore suggest that gypsy-mediated genome sites expansion in truffles follow a self-perpetuating model for heterochromatin expansion. Indeed, chromovirus-related gypsy elements are able to directly recognize histone modifications. So they have a target preference, insertion preference for heterochromatic genomic regions. Here they perpetuate the heterochromatic marks, inducing epigenetic modifications. But what about the genomic consequences? So we look at the synthetic relationship between tuber pansiwanensis and Morchella sextellata, a Morchellacea species that diverged around 250 million years ago from truffle lineages. And we found relatively high levels of mesosynteny conservation between the two species. Whereas the most important outcome of this transposon burst appeared to be an elongation of synthetic blocks due to the elongation of heterochromatic genomic regions. Indeed, tuber pansiwanensis synthetic genomic regions are on average 4 times longer than Morchella sextellata one. However, currently with the expectation that transposable elements can trigger genomic rearrangements and establish novel non-homologous genomic regions, we found higher T content in non-synthetic genomic regions, as well as lower gene density. And at the same time, we also found that genes belonging to gene families, fast-evolving gene families that experienced high duplication rates during the diversification of Tuberaceae, as well as in the Tubercuanzianensis terminal branch, are more T-rich, both in their exons, flanking regions, as well as introns. And moreover, they are also enriched in T hotspot. So it seems that we have a colocalization of fast-evolving gene families with genomic regions, with T-rich genomic regions, basically. So to conclude, we found a late Cretaceous origin of Tuberaceae, followed by a subsequent Paleogene diversification of true truffles, concurrently with the radiation of angiosperms, their main hosts. During this diversification, we had lineage-specific bursts of activity of chromovirus-related gypsy elements, and the preferential insertion of HED gypsy transposons in heterochromatic genomic regions lead to self-perpetuating elongation of these regions, driving the genome size expansion of Tuberaceae. And finally, we didn't identify any loss of mesosynteny relationship with Morkellaceae, but instead we found that the main outcome of this T burst has been an elongation of synthetic block, with colocalization of multi-copy gene families with T-rich genomic regions. I would like to thank all of you for your attention, BGE organizer for giving me the opportunity to talk at this conference, and to all my collaborators. Thank you.
Hello, my name is Pio Sierra. I'm a PhD student from the University of Cambridge, and I'm going to present how to identify transposable elements in pangenomes with a tool that we have developed, which is called Pantera. So really quickly, because I guess other presentations will also talk about transposable elements, simply to remind that they are fragments of DNA that move around in the genome and can create new copies when they do that. And the usual ways to identify them is through structural features and the repetitive character that they have in the genome. For that, there are literally hundreds of tools that focus on whether they have the sequence repetitive in the genomes, or they found similarity to other previously reported transposable elements, or they look for the structures that they have, terminal inverted repeats, long terminal repeats, etc. And we have tons of those tools. Basically, the way they work is they look for the repetitiveness, they compare the homology with previous libraries, and then we produce the annotation. But what we realize now is that we also can focus in another less studied before characteristic of the transposable elements, which is the fact that they are polymorphic. So a really brief view of pangenomes. Pangenomes, basically, in the end, they are just an alignment of several haplotypes, several sequences, in which we can find a relationship between the different positions in different loci in all of them. So in the graph format, that's one way to store them. They look like something like that. They can be kind of a mess if they are not properly construed, or in complex regions like the centromeres or very repetitive regions. But still, for our purpose, they can be really useful. Why? Because transposable elements, due to the polymorphic character, they will appear as bubbles in the pangenome that we can identify and we can collect to build those libraries. So this is an example of a pangenome in which we see here 5 haplotypes. Each of them have different polymorphisms that you identify there as a sequence that is present in just one haplotype for the case of 1, 2, and 4, or in 4 of the haplotypes for the case of 3. And actually, in this real case of transposable element of Drosophila melanogaster, we see 4 individual transposable elements for which we can obtain directly the sequence from the pangenome without needing to reconstruct it from extending the sequence when we use a repetitive method. So we have developed a tool, Pantera, that does this. This is an example of running it in a pangenome of Drosophila, and the library that we obtain is really close to the curated library that exists for this species, which is one where the transposable elements have been more properly studied. At the bottom, as an example, just a transposable element for which the LTRs, the long terminal repeats, have been completely resolved with this. And even if we need to build a pangenome, it still is really time and computing efficient. Let's say the most negative aspect is that you need to come with at least 2 or more haplotypes to compare, because it's a comparative method. So this was about the tool. Now, what we have done with this tool, I present 2 examples. One, we have created a library for cyclists of Lake Malawi that we study in our lab, several people. We have uncovered like a huge unreported diversity of transposable elements. In blue here, you can see the length of the sequences for some of the families that have been previously found. You can see some examples where we have been able to find with Panthera the actual full transposable elements that have been previously unreported, or for some ERBs, etc., even the electrons. With this really properly curated library, first we have found that many of the genes that automatic tools annotate in the genome, in the case of these species, that actually they have many different transposable elements families still active, unlike, for example, what happens in humans, many of those genes are actually transposable elements, which have been not properly annotated before. Then we use that new properly curated transposable element library to do some population analysis that we have done previously with SNPs, but to try to see what was the results that we obtained when we were looking at T polymorphisms rather than SNPs. On the left, you can see a phylogenetic tree of those clades in Malawi that we had previously studied. And on the right, you can see the PCA that we obtained from 15 different populations belonging to all these clades, and how the population structure that we uncover using the whole complement of T polymorphisms is reflects pretty accurate what we had obtained with SNPs. But we know that, we guess that the activity might be different in some species. So first we look from the superfamily level of transposable elements, and we observe, you can see on the right, that the allele frequency of this, looking at all those polymorphisms of the different families, is very different from all of them. In most cases, it's low because transposable elements are generally under purifying selection. But you can see that it's different from each family, and also for some families also there are outliers that you can see as black points, in which means that in some particular population they have a different allele frequency. For example, piggyback are elements which in general do not have a very low allele frequency, but there's, in particular, I noted these 2 particular populations where they have been recently active and they have a lower allele frequency. And that happens with other superfamilies. In the left, you can see that if we plot the number of insertions that we have in the genome compared to the number of polymorphisms that we find, we can establish a relationship with, we believe that there's a correlation between the number of insertions already present with the number of polymorphisms. We see that some of the elements will keep that relationship. These are line 2 or mariner elements. These are elements which have been active and are still active from a really long period in these genomes. They are like, you could think of them like the baseline for the transposable element complement, but we can see that families or family regs have been recently more active, same as cacti elements, this CMC. So Another way to look at it is we can build trees using the distance of the different polymorphic insertions between each of the populations. And we can see that in some cases, as I was explaining, piggyback has been more active in Astatotilapia calypterals. So they create more diversity just in this clade. This is line 2, which is the baseline again. So this is what would be most similar to the tree if we build it with the unrooted tree, we build it with all the genomes. And RT-BOB is active particularly just in the Buna clades. So all this differential activity, this is a mock-up tree that you can see. So we see how there were some families that were active already in the ancestral, but after that, after speciation, some of the families have been more active in some of the different clades. They might have been active during a speciation or after a speciation. We cannot confirm that. Another thing that we have been looking at with Panthera that enables us this possibility is that now we are having access to many more high quality genomes in our particular case. At the Sanger Institute, the Darwin Tree of Life project is generating literally thousands of new genomes. And we have been focused on Lepidoptera, for we have 455 genomes, and we created with Panthera transposable element libraries for them. So in particular, we're focusing on a type of transposable elements, Maverick, that due to the fact that it's really large, like from 15 to 20,000 bases long, and also usually present in just a very low copy number, has been probably underreported now in many genomes. So it's been found all through eukaryotes, but just in some particular cases. So we're looking at all these new genomes. In particular, we found them in 309 species, and we found 599 new Maverick families with the usual method of dividing them by an 80% divergence on 80% of the genome, which basically triples the number of transposable of polyton of Maverick elements that were reported previously in red base, which is we define the main repository of transposable element families. More interestingly, we have been focused on identifying what's the structure of this Maverick. Previously to our work, there have been like 2 superfamilies of Maverick identified, one mostly in arthropods and other invertebrates. We can confirm now that at least, for example, in Lepidoptera, all these new Mavericks, they belong to mainly 3 monophyletic superfamilies that can be divided by the structure of the proteins inside. So the ATPase in the interways, they form a nucleus, and the DNA polymerase base forms another segment, and the relative orientation of them inside the sequence differentiates and separates the different Maverick families. There's a couple of them that we don't know if they are new families that have been created through a combination, but at least in one case, this type 5 is present in just one species, and it has a completely different DNA polymerase base to others. So maybe it's a new Maverick that could have been introduced from external virus. So the takeaway messages of my talk is simply that things produce polymorphisms, and we can not only focus on the repetitiveness, the homology, or the structure to find them. We can also look at the polymorphisms to create T libraries. In our particular work in cyclids of Lake Malawi, we have identified different bars of T activity that coincide with the speciation. Now, whether that's causality of consequence, we still need to work on that. Probably it's just a correlation. And there will be new genomes and improved adaptation that will help us uncover unreported transposable elements and improve our understanding of their evolution with their host. And just thanks the people of my group and other people that helped me from different teams and the European Union for the grant. That's all. Thank you.
Hello everyone, my name is Jule and I am a PhD student at the University of Copenhagen under the supervision of Josephine Stiller. The project that I'm talking about is funded by the Willem Foundation and we are collaborating with Lucas Kodana from Illumina. It is my great pleasure to be talking about our insights into the genome evolution in signatiform fishes. As a disclaimer, all the data and the results that I'm showing are preliminary and we're still in the process of analyzing and understanding everything. But there are already some very interesting things that I'm happy to share today. So signatiformes are an order of bony fish, which are great to study evolutionary questions as they show a great range in diversity. So this is not only in morphology, as you can see in the pictures, but also in ecological and life history traits. So for example, they are distributed across the whole globe, except the polar regions, and they share their body size can vary from a few centimeters in some species to more than a meter in others. If we look at the phylogeny, signatiformes consist of 9 major families, and they share our most recent common ancestor around 90 million years ago. Also, if we look at how many species there are in each family, this is very diverse. But for now, this is not equally reflected in the amount of currently available genomes. So this is where our project comes in. We are working with a dense sampling approach to provide genomic resources across the whole order. And with our sequenced genomes, we're covering almost half of all signatiform species. So we have in total created assemblies for 242 species. As the used samples are mainly from museum specimens, we used Illumina for sequencing and then assembled with Mazurka into short read draft assemblies, which are highly fragmented, as you can see by the N50 and Busco quality measures. Based on these assemblies, we then performed a repeat annotation using a combination of repeat modeler and repeat masker, and we also built a time-calibrated phylogeny for the whole dataset. Of course, with these draft assemblies, the question arises how much of the genome size we are really missing compared to the high quality genomes. And this, with the current data, is very difficult to answer. But as there are already some published genomes available, I'm going to compare for example species how our genome assemblies look like compared to the published ones. So our genomes are the ones in green, and you can see that we compare them to chromosome and scaffold level assemblies. And if we look at how much in percentage of the assembly size we are missing compared to the existing ones, this is quite close, so within 5%. So this looks like really good evidence that when wanting to estimate genome size, our assembly sizes are getting quite close. But then if we look at a fourth example, we see that this assembly did not work really well in our case, and we're missing around 45% of the genome size compared to the chromosome level one. You can also see that this assembly is much larger compared to the previous examples, which might be causing problems. So overall, for estimating the genome size, we seem to be doing well, but there are some species where the assembly sizes are really diverging. If we now look at the first results, this is the phylogeny with all 9 families. And then for each species, we have the assembly size. And I want to point out 3 different things. So the first thing is that the assembly size is really diverse, with the smallest genome of less than 300 megabases and the largest one of 1.8 gigabases, having more than 6 times the size. And they are also from the same family of fish. The second thing to notice is there are these kind of blocks of species that have similar genome size. And this is because the genome size is highly similar between species that are closely related, as indicated by a close to maximum value of Pagel's lambda. If we compare our whole data set to what has been previously known from the existing assemblies, we can notice the third thing, that is that the existing genomes have already captured some of the increases in genome size, but are also missing others. So for example, in the highlighted family, in our data set, you can see that there's quite a lot of variation, while the published 2 genomes were very similar in size, hiding the variation that is actually there. If we look at the genome size variation in a bigger context, we can compare it to genome sizes in other groups of animals. And this plot is based on data from the Genome on a Tree database, and I have added our samples here. And if we compare to other ray-finned fishes, we see that signatiformes are on average a bit smaller than the average fish genome, but they do have a notable range for vertebrates. Then looking at the repeat annotations, it's the same figure as before, but the assembly sizes are colored by the parts that are non-repetitive in blue, the TEs in green, and the non-TEs in yellow at the tips, which is very little. And as expected, the genome size variation is majorly driven by the TE content. So if we look at the non-repetitive part of the genomes, this is very similar across the whole tree, while the TEs vary a lot. And if we put this into numbers, TEs and signatiformes make up between 6 and 75% of the genome. And we can compare this to a previous study that was done on 100 teleost assemblies. And even though they used a much broader taxonomic range, the range in genome size that in TE content that they find is smaller than what we find. And similarly, we can compare the correlation between TE content and assembly size, and we find a highly statistically significant correlation with an adjusted R-square of 0.64, and it's also in the plot, you can clearly see this correlation. And this is again stronger than the correlation found in the study on teleost in general. So the main takeaway message is that our dense sampling approach and focus on one specific order of fishes resulted in a larger variation in TE content and also a stronger correlation with the assembly size. Let's zoom in even a bit further and we look at which specific TE classes the repetitive parts of the genome consist of. And we are now looking at the TEs only and color them by the different classes. And for example, in this clade, you can see that there is a clear expansion in TEs, which is mainly dominated by DNA transposons in dark blue, while in this other clade, we have an even larger expansion in TEs, which is again an expansion in DNA transposons, but also an additional expansion in line in green. So with these TE classes, there's a lot more to investigate, but what I'm working on currently is to improve the percentage of the TEs that are actually put into one of these 4 classes. So as you can see in this plot, there's quite a large proportion of TEs that are currently unclassified in green, which is on average 50% of the TEs. And this is not unusual for fishes, and also the chromosome level assemblies in this order show around 50% TE classification only, but it might be important to look at what these 50% are before working on it further. And I'm trying some methods currently to improve this. Another thing that we are trying to understand is why there are these large variations in genome size. And in a first approach, we tried to look at it from an evolutionary perspective to see how the traits evolve over time. So we took the tip values and then we reconstructed ancestral states through time. And what you can see here is how the genome size evolved. And there is this clear period during the Eocene where the assembly size increased. And if we look at the same plot for the repeat content, we can see that during the same period, this also increased. And interestingly, if we look at the GC content in the same period, there is a decrease, which might make sense in a way that repeat-rich regions tend to have lower GC content. Then the genome size variation could also be further understood by looking at the correlation with other traits. So here are some example traits that have previously been shown to be associated with genome size in other groups. And in a huge effort, we tried to compile all available data for these traits in our group of fishes. But first investigations do not show such a clear correlation. So then to summarize our insights into the genome size evolution in signatiform fishes, we have found that signatiforms have evolved a huge variation in genome size, and this range is akin to the range reported across bony fishes. We also see that TEs are the major drivers of this genome size variation, and that there are multiple expansions of genome size via roots of different TE classes. Thank you very much for listening, and I'm very, very happy to receive any kind of input, suggestions, or questions.
Okay, hello everybody again, and welcome back. Thanks to all our speakers for some really great talks. We heard work for across very wide diversity, faces fungi, arthropods. And I'd like to start with some of the questions from our audience. On your questions, okay, we will start with some questions for Marta. I think there was a question that came up a couple of times from both Marta and Mark. The question was, are there any shared genes showing signatures of association with urbanization due to parallel evolution or hybridization or introgression? Is this something that you've seen in your data?
Thank you for the questions, Mara and Mark. And indeed, during that time that I recorded the talk, and today, that was a month, I've been working on this, on detecting integration. I am not using the haplotypes, but I am applying the ABA-BABA test. And it's still working in progress, but this is something that we expect also to detect. So, yeah.
Okay, thank you very much. So maybe we'll go on to another question, again for Marta. We'll go in a row here. So did you actually model the likelihood of random association between genes and TEs? Is it something that you've looked in your data?
No, I have not, but thanks for the suggestion, because we will apply this model.
I think that would be quite interesting.
Yeah.
For your type of data as well. Thank you very much, Marta.
Thank you.
So then we have a question for Pio. In order to run Panthera, the genomes that you're using for the pangenome, is there some quality level that you're suggesting as necessary? People are asking.
Well, more than the quality level, that of course the results that you will obtain in terms of the consensus sequences will be related to the quality level. What you definitely will need is a certain level of identity between the different genomes that you use. That is, you just can align genomes which are closely related. They, in some cases, they don't need to be from exactly the same species, but they will need to be from closely related species enough that you will have enough synteny to create a pangenome.
Would you, do you have like a possible cutoff? How many million years if you've tried something like this?
Well, I would rather not use years precisely because with transposable elements, we see how the part of the whole pitch is that the diversity created by excess of activity of transposable elements can produce a larger diversity between different genomes in a shorter span of time of which we would usually measure using nuclear mutation rates, neutral mutation rates. So what I would say is something around at least 90% of identity, 93 is necessary to build them.
to be able to build the pangenome. Okay, great. Thank you very much, Pio. Let's see. Actually, I have a question for Jacopo's talk. I think you mentioned some lineage-specific expansion, especially and with gypsy retrotransposons. Was there a biological cause behind that? What do you suppose is the reason for that?
This is a very, very good question. And I would like to have a clear answer for that. And probably there are different evolutionary forces that are acting, like demographic history of species. It's big evolutionary transition, having a hypogeous fruiting body. So maybe there are some changes in effective population size during the establishment of this phenotype, and also the radiation should have been quite fast of truffles. So probably this expansion started in their most recent common ancestor, but what we can see now is more related to lineage-specific amplifications. Also because we have seen that non-allelic homologs recombination is acting in the genome. So we have deletion events. So it's possible that more ancient shared transposition events have been removed from the genomes, basically. And so we are looking at the process more lineage specific. Yeah. So yeah, I don't have a clear answer. Yeah.
Well, it sounds quite interesting in this particular case. Okay, thank you very much. Let's see what else. We have more questions for Pio. Okay, one question from Josefa. Have you estimated how the number of genomes included in the pangenome affects the number of Ts? I guess how many genomes?
No, I have some data, but I didn't include it. Yes, so of course, as you add more genomes to the pangenome, you will include more polymorphisms, and you will identify new insertions. And also, I would say even in some cases, one of the things that we find with this new method is that you can find insertions on some elements, particularly large elements like large endogenous retrovirus or these maverick elements, which are very low copy number. On top of the complexity of including them due to the size, you will have just one or two in the genomes. So in those cases, the more genomes you add, the larger the probability that you will find them and not think that they are not present at all in the species.
I see, I guess. Okay, that's good. I think that also covers Mark's next question on finding viral insertions, which I think you just covered as well here. Maybe we can have a question for Yule. The genome size plot through time is really cool. Me try saying it would be interesting to know which T families were contributing to that. Did you find any specific families?
I have not looked at that yet, but that's a really interesting thing to look at next for sure. This was just all the TE content put together that I showed, but I can definitely plot the same thing for the different classes and see which one fits the best or shows the strongest correlation. Yeah.
Again, for your correlation analysis, Mark is asking if you correct for phylogeny based on the phylogeny.
Yes, all the correlation analysis have phylogenetic correction in it, yes.
Okay, thank you very much. Some more questions. Did you look at the genome size estimate using K-Meyer spectra?
Yes, so we did KMC, and then we kind of compared that those results are similar to what we get for the assembly sizes from the assemblies. And we also did the same for the repeat content to make sure that we are not super far off. And that also correlated quite well. But it might be worthwhile to also do some of the analysis that I did now based on the assembly sizes with the estimated.
Exactly, that would be interesting.
Yeah.
I think we have a similar question as what talked about taxa proximity here, based, analysis based on with phylogeny. I think that you discussed earlier, and let's say another question for Pio. Did you identify Ts that were not varying across the genomes with Panthera?
That were not what?
How do you identify transposons that are not varying across genomes? So quite
No, you will fail to identify them. Any transposable elements which is not polymorphic between the genomes, you will fail to identify. That's also why having more genomes in the pangenome, as it was earlier suggested, it will increase the identification. Also, that maybe I didn't explain it completely earlier. There's a bias in the sense that you will be able to identify most recent transposable elements, that is, those which have transposed more recently. They will have lower allele frequency. Those are the ones that you will most likely identify. Whereas all transposable elements whose transposition happened hundreds of millions of years ago, they might be fixed completely. The only remains that are left, and those will not be identified at all by Panthera.
Good. Thank you very much, Pio, and thanks everybody. That was very interesting. I think there's still a few questions in the chat. If you can contact the people directly. I think we're just running out of time now, so I'm going to wrap this up. Thank you very much all for your interesting talks. And for the audience, please stay along for the poster session, which is the next session coming along after us. And have a good evening.
Thank you, Elena. Thank you.
Thank you.